Bismillahirrahmanirrahim. Let's see how different biosensors can be used to detect the presence of antibiotics in the environmental samples. So as we know, antibiotics are of major concern, and these are can be and are life-saving components. And however, the unchecked use of these antibiotics is obviously not good. It can give rise to the microorganisms which can be resistant to these antibiotics. So we know that medical substances, antibiotics and related substances have been released in the environment for years. In some areas, in some countries, there is no check and balance how to dispose of these substances, and therefore the presence of these antibiotics in the environment has raised certain concerns. Obviously, when the microorganisms are exposed to antibiotics and the prolonged exposure, if the exposure is towards against a high concentration of antibiotics, obviously the bacteria will die. But if the concentration is not high, not all of the bacteria will die. Some will survive, and the surviving bacteria will develop mechanisms and strategies to fight against these antibiotics. And these strategies are basically based on antibiotic resistant plasmids, and these plasmids can be transferred between bacteria from one bacteria to another, which is called as horizontal gene transfer. So if these plasmids which are carrying antibiotic resistant genes, if these are transferred from one bacteria to another, it means the resistance against antibiotic is spreading. So therefore antibiotics exposure to bacteria or to normal flora should be only done when required and not occasionally. So if these are released in the environment, obviously the bacteria present in the environment will develop resistance against antibiotics. This is of concern because these bacteria can ultimately find their way to life as well. Moreover, antibiotics are given to dairy cattle and the farm animals which are reared at farms, fish as well, and livestock. And this unchecked antibiotic exposure has also given rise to so many different bacteria in these animals which can now resist different types of antibiotics. And if these bacteria find their way directly or indirectly to human beings, it means those antibiotic resistant genes are now being introduced to the normal flora of the human beings as well. So that is not good for the health of human beings, because it can give rise to so many different multi-drug resistant bacteria. So it raises a significant question about the food safety, that how good is the quality of the food. Antibiotic resistant bacteria or other such molecules present in the food are of concern as well. There have been a lot of research to find out the presence of antibiotics and related compounds in different environmental samples and food samples as well. For example, a commercial biosensor Biocor 3000 was used to study the cross reactivity between two sulfonamides, sulfamethazine and furosemide. So there are different types of these compounds, and these can cross react as well, which is again not good for health. Sulfonamides sometimes can cause allergic reactions, and the effect in human immune system is of high interest. So there have been construction for so many biosensors that can detect these compounds. Another example is of whole cell biosensors, which is based on 3 reporter genes from 3 different microorganisms: Vibrio fischeri, Escherichia coli, and Escherichia victoria. So different genes were taken from these different microorganisms and were combined together to make one biosensor. It is called 3 reporter gene system. It was combined with tetracycline inducible promoter. So you can see here, when tetracycline inducible promoter is there, it means this promoter will be activated only when the tetracycline is present, because it is behaving as an inducer. So if tetracycline is present, the promoter will be induced and it will start transcribing these marker genes. So this biosensor was reported to respond to even low levels of tetracyclines by producing different types of signals, for example galactosidase, light, or even green fluorescent protein as well. So these are a few examples how different biosensors can be used to detect the presence of antibiotics in different samples. We'll talk more about the application of biosensors in our next lessons.